Hello everyone, my name is Conan Lee, and I am a biochemistry major at UC Davis. As an undergraduate researcher, I perform research at UC Davis at the Atsumi lab. This is our current world, and every day, we use products such as plastics, cosmetics, fragrances, and detergents, and they're all derived from the petroleum industry. The petroleum industry produces a lot of pollution, as well as hazardous chemical waste. Pollution is bad for both us and the environment, and therefore the Atsumi lab is trying to come up with an alternative to reduce this dependency on the petroleum industry and therefore produce less pollution. How do we do that? Our first step is using plants. Plant biomass sequesters carbon dioxide and converts that into sugars. We can extract that sugar and feed it to specifically engineered organisms to produce our products of interest. And here, we're using microorganisms as a means of production systems. So how does this system work? It works through the process of genome engineering, in which we're essentially installing a gene that enables a host organism to produce the products of interest. And here, we're working with E. coli, as it is an extensively studied host microorganism, and it gives us flexibility in the research lab. And currently, we're trying to synthesize isobutyl acetate. It is a versatile ester chemical compound. For example, it's used as a precursor to a drop-in biofuel candidate, as well as a solvent in various food flavorings, fragrances, and paints. And currently, we have a pathway to make this, in which it involves both the natural pathways in E. coli, as well as synthetic pathways that we've installed. In the laboratory, when we were trying to make isobutyl acetate, we ran into a problem. We actually found that IBA is toxic to E. coli, and this is bad because it's going to inhibit the amount of production that we can make. So how do we fix this? We can fix this through a process called directed evolution. And essentially, we're inducing mutations and for a higher tolerance. And here, we're using a selective pressure of isobutyl acetate, and we're reintroducing mutations throughout a constant cycle. With this mutant strain, we're actually able to have a higher tolerance as well as a higher amount of production of isobutyl acetate. And now that we have these mutations from the directed evolution, we need to find them. Well, what can we do? We can do sequencing. We received funding from the Joint Genome Institute to perform sequencing, and we collaborated with Dr. Elias's lab to analyze that data. And from that data, we found that there were 6 integral single nucleotide mutations, 5 of which were related to the membrane, and 1 which was in the DNA structures. So now that we have these mutations, we need to assess them. Since we have 6 mutations, we actually have a total of 63 combinations, and therefore we need to test each combination to see what works with each other. And our future goal is to essentially have an increased production and tolerance of isobutyl acetate for the best combination possible. And of course, we want to have an impact to our current society and environment. I would like to thank you for watching, and I would also like to thank everyone in the Atsumi Research Group. Thank you.